Assalamualaikum. Myself, Tania Tilbar. I am a student of microbiology at KUST University. My topic is screening of selected medicinal plant extracts against Streptococcus pneumonia isolated from pneumonia patients. Now, how this idea came in our minds. This idea is particularly very different, and these medicinal plants such as Dricata and Neligica and Acarus and Pispera are very necessary against these diseases for Streptococcus pneumonia. Introduction. Through World Health Organization in 2015, pneumonia is a form of an acute respiratory infection. Acute respiratory infection, which cause lung inflammation, making breathing painful and limiting oxygen intake. Pneumonia is a common occurrence after stroke and contributes to the morbidity and mortality experienced by stroke survivors. It is estimated that 6 to 22% of survivors will experience such against for pneumonia. Through Marianne et al. At 2013 described this Streptococcus pneumonia frequently colonize the human nasopharynx, but can cause severe disease fit and admits normally sterile sites. Bacterial infections, which are resistant to antibiotics, can limit the availability of effective treatment options, rendering some commonly encountered bacterial infection to treat. The bacteria, then we use some herbal medicinal plants which are resistant which are resistant against Streptococcus pneumonia. Herbal drugs are the potential source of therapeutic aid for the treatment and prevention of number of disease. It is documented that herbal plants are very bioactive compounds. The rich biodiversity of plants make them a treasure housing for obtaining new and novel compounds, either as a drug or lead molecule called drugs with different mechanism of actions. Study objectives. Here, we have three main objectives for this research study, such as first, to isolate the antibiotic susceptibility test against ammonia toxide from hospitalized patients. To screen second, to screen the selected medicinal plant extract against S. pneumonia, Streptococcus pneumonia, to determine the MIC of medicinal plant extracts. How these why these problem has why these problem are more severe. Pneumonia is one of the most prevalent opportunistic pathogen. This study aims to isolate the antibiotic resistant pneumococci from these high specialized patients and screen the selected medicinal plant extract against this Streptococcus pneumonia for determining the MIC of medicinal plant extracts such as Escherichia indirecta, indica, nilotica, and Acarus inspira. These medicinal plants have not previously used against Streptococcus pneumonia, so we have applied these medicinal plants which we identify, which we identify if these plants are if these plants are mm, uh, effective against Streptococcus pneumonia or not. Previous literature. Weiser et al. At recent research in 2018 described that the Streptococcus pneumonia is one of the most opportunistic pathogen. This bacterium colonizes in the human nasopharynx, which make colony and cause severe disease such as meningitis, uh, such as meningitis, also in problem in intake of oxygen. Pneumococcal disease, including pneumonia, sepsis, and malaria, lead to millions of deaths per year. Also, antimicrobial resistance is also in antimicrobial resistance. It is documented that now it is key for the health, uh, which were described as Hutner et al. In 2013. Community-acquired pneumonia is a common infectious disease with a significant mortality rate, although various microbial infections are associated with this disease. And then material and methods. Through experiential study, we choose the population of district correct. We collect the sample size and 45 hospitalized patients. We make consider ethical form. Through this ethical form, we collect the data from hospitalized patients. We collect the sample of hospitalized patients, and then we apply the antibiotic. Then we apply the antibiotic susceptibility test. Then we collect the local medicinal plant collection, which are not used against this disease. uh, uh, we collect some of the plants such as the Isida dicta, Nilotica, Venicans spera. We make these plants, we make extract of these plants and different solvents, organic solvents such as methanol, ethanol, and also in distilled water. Then we apply the antibacterial activity through either well diffusion method. We check the antibacterial activity of we check the antibacterial activity of different antibiotics, and then we identify which antibiotics are more resistant for Streptococcus pneumonia. And the last step is the determination of MIC. And results. First of all, culturing growth of bacteria. For culturing the growth of bacteria, we prepare a media and water for the growth of bacteria. After that, the media is prepared. We sticking or putting in the sample of hospitalized patient, and then incubate in the and then incubate for 24 hours. Then we check if the growth are occur. Then the second step is gram staining. Streptococcus pneumonia is a gram positive, streptococcus gram positive, and diplococci. And this is one of the image of Streptococcus pneumonia, which is diplococci. The the third step is biochemical test. We have different tests to check the Streptococcus pneumonia, and I have to choose some of the tests such as catalase, oxidase, urease, and one of the tests is catalase test, which are negative for the Streptococcus pneumonia. And the fourth step is antibiotic erythromycin. We have applied different antibiotics. One of the antibiotics and some of the antibiotics such as erythromycin and dimethyl sulfoxide. And this image erythromycin antibiotics is show a resistance against Streptococcus pneumonia, which is seen clearly in this image. Conclusion. In overall study, it is concluded that sample extra antibacterial activity compared with standard drug azithromycin and dimethyl sulfoxide showed resistance against Streptococcus pneumonia. The antibacterial activity was determined by measuring the zone of inhibition visually in patients. Recommendation. It is recommended to the policymaker that these medicinal plants, flora such as indica, luteca, and Achairus infespera, but in the Streptococcus pneumonia, so more and more prefer to these plants for the given in the next for the future. So this is references, some of the references we can. Thank you for listening us.